So we're now going to cover the HL content, which is luckily quite brief for this. So first, identify and describe the cells that increase surface area to volume ratio. So they've specifically said what cells increase this ratio. There are a few, but just think about this real quick. So we have your red blood cells or erythrocytes. Let's write that. Erythrocytes. And then we have our proximal convoluted tubules. So proximal convoluted tubules. Then finally we have pneumocytes. Sites I and II, so 1 and 2. All three make use of the cell surface area to volume ratio that we discussed in the previous video, specialization regular, standard level, to make the best use of it to do their metabolic function. So let's actually look first at erythrocytes, since it's already up there. What is an erythrocyte? Well, it's a red blood cell, pretty simply put. It looks something like that from the front. So it's 7.5 to 8 meters / 8 micrometers. It has a large concave disc kind of shape. So it's inside, that's from the, there you go. Looks something like this. And if we just look at it, just define it as 1 unit here, 1 here, so a total of 1 unit and a total of 2 units. So just think about this. It's actually going to have a startlingly large surface area. So even if this was a square, its surface area, so SA, surface area, is high, volume is low. Now, why is that important? Well, this allows for gas transport, so O2 transport. It also allows it to maximize O2 transport. That's also why it doesn't have any cell organelles, because it really doesn't need it, right? It only has one role, and for that role it has no need of it. So the volume is low because it has no cell organelle. So this is the first one. The second one was the proximal convoluted tubule cells. So let's actually write that. Proximal convoluted tubule cells, and those are actually within the kidney, somewhere here. So let's actually think about what these are. Draw this. This is a very simplified drawing. So they're responsible for reabsorption of nutrients in kidney, e.g. glucose, right? And this is going to be a little tube, right, carrying the glucose. So let's say this is glucose that we represent by these little dots. Now this tubule, a cell, has microvilli here. So in the exam, do not draw that. Draw the arrowhead, so I'm just gonna omit it here. So this is microvilli, and this allows for increased surface area. So increased surface area, and that speeds up rate of absorption. So that speeds up rate of absorption, because it actually diffuses faster. So this part of the cell has a slightly lower surface area to volume ratio, but this part of the cell, let's actually draw it like that, so it looks like it's on 2D, right, has a greater surface area to volume ratio, which means that it's going to absorb things faster. And that's the main use. Now let's move on to the final one, the final example, which is going to be pneumocytes. Or we can just talk about the lungs in general. So now we will talk about just pneumocytes. So the alveoli, let's talk about, so the alveoli, actually, that would be more apt. So the alveoli. Now the alveoli will look a bit like broccoli, definitely. Let's get that. All right, but ignoring the interesting drawing, they have a very porous, or like a open structure, and that allows them to have a very high surface area to volume ratio. So let's actually draw a slightly larger diagram of the alveoli, like that, right? And really investigate what's happening. So there's a little bit of diffusion that happens here, right? So O2 comes in, CO2 is carried out. But what we're really looking at is the cells within that, cells that comprise, the alveoli comprises of, and that would be, in green, are type 1 pneumocytes. So we're gonna label it like this, right? So all the green, including the little dots, like this is the area, right, are going to be pneumocytes. They cover about 95% surface, and their role is to allow gas exchange. So their role, I'm going to write this in red because it's important, is gas exchange. So that's why they have an extremely large surface area. And here we're going to have our little capillary, and that's actually going to have the diffusion happening. Additionally, we're going to have type 1 pneumocytes, which I'm going to draw in red, even though I wrote something in red, so that's a bit confusing. So yeah, so I'm going to erase this and actually rewrite it in green so that there's no confusion. Gas exchange, and they're on the surface. Type 2 pneumocytes are actually going to be in here, within this. So let's draw them like that. So pneumocytes 2 would actually be 5% surface, and they used to secrete surfactant fluid for gas exchange. And that's pretty important because that just allows let's just say for diffusion. That simplifies it a bit. For diffusion. And this is gonna be the main the main three types of things. So you have your type 1 pneumocytes, which is lots of coverage, and they are for the gas exchange, for diffusion. You have your type 2 pneumocytes, which is smaller surface area, but they have an equally important role, and they both are within the alveoli. So these are going to be your 3 cells. And yeah, now we're going to move on to specializations of a couple different things. Now, we just spoke about the alveoli, and that is within the lung. So we're not going to talk about that again, but on the list of things to discuss, we actually have the lung, the heart, and the muscle, as well as gonads. Gonads are sexual organs, so testes and ovaries, sorry, right? So we already spoke about the lungs, we're gonna cross that off. Let's talk about the heart cells. So the heart, just like the muscle, the heart is a muscle actually, has striated muscle cells. And actually it has cardiac muscle cells. And they have these things, let's draw a little structure like this, right, called contractile heart. Let's put this here, just that. I need to know how to spell this. Contractile microfibrils. Yeah, so contractile microfibrils. So what is that? It's an extremely long word. Well, let's draw that. A little bridge here maybe. And this is gonna have multiple nucleus because again it's a muscle cell, so we already know those can be extremely long. So let's write that cardiac muscle. So, all right, now within the cardiac muscle cells, the interlaced discs, which we're gonna draw here. So these little discs all have a specific function. So we know that all muscle cells are kind of special, mostly because they have a different structure, and that's because they have they're all uninucleated, which means all the nuclei are within one cell, and that actually comes really in handy because you need to produce a lot of energy in your muscles, and that's basically the most important function. Now, the cardiac muscle cell depicted up there has a slight specific specific use. So the I don't know how to spell that. So the intercalated discs. So let's actually go here. Intercalated discs. Label that in black. So the intercalated discs are actually branching here, and this space allows ion movement. So in this case, the specialization is that the cardiac muscle cell has these intercalated discs, and they have gaps between them to allow the ions to move in and out. This differs slightly from a regular muscle cell. So this would be called, this would have a branched nature, let's call it. Branched nature. There we go. While a regular muscle cell would actually just have a fibrous nature. So this is important because it has to rapidly, let's actually write this. So in red, very important, rapid expansion slash contraction from electric impulses. And basically this is extremely important because this allows the structure allows it to actually do this, right? And the contractions are important because if it was just a very long strand, it would still be able to do that, but with less flexibility. So this mesh-like pattern allows that flexibility. So this is a heart or cardiac muscle. So just take that, let's erase this and move on to regular muscles, striated muscle cells. Muscles, striated muscle cell. All right, now striated muscle cell, very similar to a regular heart cell. Let's draw that like that. Is long, but it's also more fibrous. It doesn't have those branching patterns, but it does have the discs, the intercalated discs. Again, it's going to take a little bit to draw. But what's important here is to note that there are a lot of similarities because they're both muscle fibers. And I use the word fiber here instead of muscle branch fiber or whatnot because it is just a long fiber. So fibrous slash long, up to 300 millimeters actually. So that's about 30 centimeters if we break that down, because a millimeter is a tenth of a centimeter. And we don't know a few things. So firstly, are they really cells? So should they be cells? They be cells. Since they actually have multiple nucleus, like the heart one, and they have a different structure, and there's a lot of differences between irregular cells. They actually form a single cell because of fusion of cells, so fused cells. So should it be a cell or should it be a tissue? Because a tissue is multiple cells fused into one, or multiple cells with the same or similar function. Finally, they have just like they're all along multiple fibers. So if you just think about a muscle, they're gonna look something like this with a lot of intertwining kind of, like all of these are individual muscle fibers, and they're gonna make, say this is the shoulder, right? So this is the shoulder, this is the elbow, this is the arm, this is your fist, right? I think that's 5 fingers, 1, 2, 3, 4, 5, yeah. So all of these cells are long fibrous muscle cells. So that's about it for the muscles. Let's talk about gonads. Gonads. So gonads are sperm / egg cells, and the gonads aren't those are just the cells that are in the gonads, but it's the basic outline. Let's first talk about the sperm cell. This is a labeled diagram, should do it. That. Alright, so what do we have inside the sperm cell? Let's actually draw little things here, because that is quite important. So the width is not that much, right? So it's only about 2 micrometers, but the length is more substantial. It's about 55 micrometers. This is called the acrosome, acrosome, and that's actually a penetrative head filled with enzymes that break down the barrier around the egg cell. This is obviously the nucleus, and these are, well, it's a bit odd from the diagram of the drawing, but mitochondria. Why are there so many mitochondria? Well, that's because he's a swimmer. He needs a lot of energy to actually make it up the fallopian tube to the egg and then to penetrate the egg. So the adaptation here is that his surface area is his volume is quite low while the surface area is quite high. The surface area in this case doesn't really matter that much. It's a very aerodynamic shape, and this allows for motility. So motile, energy efficient. So this is the main benefits of a sperm cell. An egg cell, on the other hand, is a little bit different. So this is a sperm. Let's cross off sperm and just talk about egg cells now. It's a bit bigger just to start off. You're gonna have a thing like that. And let's actually just look at the names of this. Cytoplasm. So there's a little matrix around this. So I'm going to represent that like this. So this is a protein matrix. Let's name that protein matrix. I.e. zona pellicula, and that actually hardens after fertilization. So after this is going to be the nucleus again, 23 chromosomes. The zona pellicula actually hardens once the egg is fertilized, because they don't want more sperm cells penetrating into it, and the zona pellicula, let's draw this here, requires enzymes to break. Like initially it requires enzymes to break, which is what is actually inside the acrosome. So if we just draw a sperm cell here, let's just draw just the head of a sperm cell. That'll look odd, so that's not. So this is going to be enzymes, right? And that actually breaks into that to fertilize it. So those are the adaptations. So the zona pellicula, as well as the acrosome, and a lot of the tail, and a lot of little mitochondria. So this should be the end of specialization B2.3. And yeah, that's about it.